Our case involved an ICU patient with fever, an inflamed peripheral venous catheter, and a positive urine sample. The question arises whether our patient could suffer from a UTI or rather a PVC-related bloodstream infection. Let's start with UTIs. Every one of 2 women will suffer from a urinary tract infection in her lifetime. In men, the incidence is much lower at only 1 in 20 due to the anatomical differences. 35% of all nosocomial infections are UTIs, making them the most common. Regular urine flow is the most relevant defense mechanism as it simply flushes pathogens away. An impaired clearance increases the risk of a UTI. Immunosuppression of any kind is another major risk factor for recurring UTIs. And finally, indwelling medical devices are an optimal scaffold for bacteria to form biofilms. E. coli is responsible for the vast majority of UTIs. However, they can also be caused by non-commensal like Pseudomonas aeruginosa. A dynamic balance between defense mechanisms and bacterial growth keeps the proximal part of the urinary system sterile, and any disturbance of that balance can lead to an infection. If pathogens reach proximal parts, their endotoxins start triggering a local inflammatory response, causing the cardinal signs of a cystitis, pollakiuria and alguria. If the infection is not successfully controlled at this point, a further ascension of the infection to the kidneys can occur, which then results in pyelonephritis. The involvement of the kidney pelvis is usually accompanied with the onset of flank pain and in systemic inflammatory signs like fever. In 40% of the UTIs with pyelonephritis, an invasion of the bacteria into the bloodstream occurs, which can lead to a life-threatening urosepsis. Changing in perspective, 25% of all nosocomial blood infections arise secondarily from a UTI. For that reason, blood cultures are usually taken from patients with bacterial pyelonephritis. Indeed, BSIs are a relevant complication to occur during hospitalization, making up 15% of all nosocomial infections. But UTIs are only one possible origin. Blood catheters are another relevant source, being responsible for every 1 in 10 BSIs. Moderate inflammation around PVC insertion sites occur fairly often, while statistically only 1 in every 1,000 lay PVCs result in a BSI. That doesn't mean that an infected PVC should be completely ignored as a possible cause of a suspected septicemia. Staphylococcus aureus is by far the most common cause of BSIs, followed by Pseudomonas aeruginosa and coagulase-negative staphylococci. PVC-related bloodstream infections usually start with a minor erythema or grade 1 phlebitis. When opportunistic pathogens form a biofilm on the catheter tip, a small amount of bacteria constantly gets released into the bloodstream. These bacteria are usually eliminated fairly quickly. However, in some cases, a local PVC infection can disseminate and cause a life-threatening generalized bloodstream infection. So now, let's turn to our case.
Our patient is recovering in the intensive care unit after recently experiencing a heart attack. Being hospitalized for 5 days with a urinary and peripheral venous catheter, the patient has developed a fever and inflammation around the catheter insertion site. Clinical data was collected to identify the causative organism of the symptoms presented. A urine sample was taken from the urinary catheter, as well as a blood culture and wound swab of the area around the site of infection being collected. Microscopy testing of the urine sample revealed biomarkers indicative of a urinary tract infection, such as high white blood cell count of 10 to 100 times 10 to the 6 leukocytes per liter. Microscopy also found there was to be less than 10 to 100 times 10 to the 6 red blood cells per liter and 10 to 100 times 10 to the 6 epithelial cells per liter. Red blood cells within the urine of this concentration is typical of microscopic hematuria, but is unlikely to be of significance in this case. The high presence of epithelial cells in urine is likely caused by sloughing of epithelial cells within the urethra, bladder, or kidneys, which is also consistent with UTIs. The culture sample should be disregarded due to the likelihood of contamination from using an invalid collection method and the inconclusiveness of the clinical notes provided. Samples acquired from a urinary catheter are not usually accepted due to the high probability of it being contaminated. UTIs typically grow in monomicrobial cultures or in a mixed culture with a predominant organism. Given the notes provided, there is no clear indication of a predominant organism within the culture, so while a concentration of 10 to the 8 would be clinically significant, it is likely that the culture observed was affected by contamination. This finding casts doubt on whether the aforementioned epithelial cells were a production of a UTI or of contamination. With no further biological data revealing the frequency of the catheter bag changes and to grow to the concentration in which the presented itself in, the bag likely not changed during the duration of the patient's recovery. As such, another sample will be needed to confirm the patient has UTI. Testing of the blood specimens found Staphylococcus epidermidis to be isolated in 2 of the 4 bottles of blood. It is possible that this is the result of contamination due to the organism being part of normal microflora of the epidermidis, but being present in half of the blood bottles, this is unlikely. With the clinical notes in this context, the organism may be a potential source of infection, considering the relatively high occurrences on nosocomial infections concerning this particular pathogen. With no pus from the wound to test, the gram stain was able to identify occasional gram-positive rods and occasional gram-positive cocci. Our culture identified these organisms as a mixed growth of diphthoroids and Staphylococcus epidermidis, microfloral organisms of typical contamination. The wound swab was therefore considered to be inconclusive.
Despite the inconclusive samples, a new workflow can be established from these results to provide a future result of the cause of illness. Firstly, the suspected urinary tract infection would be addressed by obtaining a new midstream urine sample, which is the second third of urine. This avoids contamination from the catheter urine and is a sample without urethral colonizers. From here, the workflow is as follows. The macroscopic and chemical analysis would be completed if they had previously not been recorded. A microscopic analysis is used to do a cell count to spot at any abnormal levels of cells present in the urine, and the samples need to be cultured and then completed. Any significant growth will be gram stained, and antibiotic testing and further identification methods will be used on the cultured species to help identify the pathogen. To identify the organisms responsible, MacConkey agar, horsebutt agar, and chromogenic urine plates should be inoculated. A lack of growth of organisms is typical of normal urine, and a pure culture or mixed culture with predominant organism growth exceeding 10 to the power of 8 CFU per litre is generally the criteria for a UTI. In the new sample, we would expect to see one of the 3 previous pathogens present, which can be differentiated on several characteristics despite being gram-negative rods and all catalase test positive. E. coli and P. mirabilis are both oxidase negative, thus making them a part of the Enterobacteriaceae family, which is easily identifiable through an API 20E test. Pseudomonas aeruginosa is identified in its inability to ferment lactose, a positive oxidase reaction, and its ability to grow at 42 degrees Celsius. Fluorescence under ultraviolet radiation helps in early identification of its colonies and can further be identified using an API 20NE. The P. mirabilis can also be identified through its ability to ferment maltose and inability to ferment lactose, and has a clear and iconic swarming motility formation. The recommended treatment for UTI is a course of antibiotics. Which course it will be is identified through antibiotic sensitivity testing and is completed in the lab. With the nature of the blood sample previously outlined, the best workflow to identify whether the Staphylococcus epidermidis is contamination or not would be to take multiple other samples of blood cultures. The wound swab is also concerned with possible contamination, as the Staphylococcus epidermidis is a part of the natural skin flora. The best workflow from this, alongside the secondary blood cultures, would be to culture the catheter tip and identify if the organism is present on the indwelling device. Recent streamlining of the testing of epidermidis can now make it separated from the other 46 coagulase-negative Staphylococci species through its ability to ferment mannose and maltose while not fermenting mannitol and trehalose, while all being susceptible to novobiocin. If the results from the proposed workflow are positive, the nature of the infection is to create a biofilm around the catheter and can make it very hard to treat. However, the current and best treatment response is a combination of rampicin and vancomycin therapy, as well as removing the indwelling device to restrict biofilm growth.
The processing of a new MSU sample would adhere to the following methodology. The containing bottle would ideally be labelled, refrigerated, and observed macroscopically. The colour and consistency of the sample is suggestive of infection, and as such, any observations should be noted. A cloudy pale colour, for example, is suggestive of pyuria, a biomarker for a urinary tract infection. Biochemical analysis is typically the next course of action in pathogen identification. This is typically achieved by a dipstick. A dipstick is a rapid testing product for various biomarkers associated with the infection of the urinary tract. Among these biomarkers are the pH and specific gravity of the sample and the concentration of glucose, proteins, leukocyte, erythrates, erythrocytes, and nitrates. Microscopic analysis of a representative amount is performed. A cell counting chamber is inoculated with the sample, is used to count the chambers for leukocytes, erythrocytes, and epithelial cells, also taking note of any bacteria, yeast, crystals, or protein casts observed. Keeping in mind that urine is a sterile biofluid, observations of any organism is highly suggestive of infection. Observation of yeast would warrant inoculation of a Seborrhoeides dectos agar and chromogenic yeast plates for confirmatory testing. Direct antibiotic sensitivity testing will be performed using the UCAST method on the predominant organism as a means of surveying potential treatments. These antibiotics are commonly used in treating UTIs and can be useful in identifying the responsible pathogen. Sensitivity can be ascertained by measuring the diameter of clearing surrounding each antibiotic in a lawn plate. If the clearing is less than the index, the organism is considered to be resistant to the antibiotic. If the clearing is greater than the indices provided, it is considered to be sensitive to the antibiotic, thereby identifying a potential treatment method. If a gram stain identifies the pathogen as a gram-negative rod, API 20E will be performed and a secondary antibiotic test may commence with these antibiotics for general Enterobacteriaceae identification. The API 20E can be used to identify specifically the cultured organism. Coagulase, nevabiocin, and Lansfield grouping may also be deemed suitable based upon the findings of a gram-positive cocci in the gram stain. S. epidermis is a part of the microflora of the surface of the skin, but considered to be an opportunistic pathogen. These organisms can colonize pathogenically when the biochemical conditions are in their favor. In this case, the opportunity may have been the insertion and lack of sterilization of a peripheral venous catheter. With its isolation in 2 of the 4 blood bottles, it cannot be disregarded as contamination. The novel approach by which the ailment could be confirmed as a blood infection would be to isolate the same pathogen in the site of infection. Wound swabs are generally considered to be unreliable due to potential contamination by microflora, particularly in the absence of pus. Furthermore, this process increases the risk of specimen damage due to air exposure and the risk of drying out. The most appropriate measure would be to have pus aspirated by a syringe in sterile conditions. The specimen would then be inoculated in horse blood agar in aerobic and anaerobic conditions and mannitol salt agar. If the patient is indeed suffering from a blood infection of S. epidermis, we should expect to see the growth of pink colonies on mannitol salt agar and white colonies on horse blood agar with gamma hemolysis. A gram stain should reveal gram-positive cocci and a coagulase test should present negative. Direct UCAST antibiotic testing will then be performed to identify sensitivities.
Careful analysis of the clinical notes reveals several shortcomings in the testing methods and provided a lack of conclusive results. While the concentration of leukocytes within the urine can be suggestive of a UTI, the specimen is too contaminated to draw valid conclusions on the current data alone. And the 2 positive blood polls neither allow us to conclusively diagnose nor deny a present infection. While there is a considerable amount of testing to be performed to isolate the causative organism for the fever, some immediate actions can be made. This includes, but is not limited to, changing all catheters as frequent as reasonably as possible while monitoring random urine and blood samples.